Time you're ready, we're ready.
So I'm gonna welcome everybody. Next week, who's the speaker?
It's the one on psychedelics.
Paul Hudson.
Paul Hudson, School of Pharmacy,
We have-
psychoactive substances.
Okay, so is everything, this is one that Paul was hoping for, and we've got the director of the new center that was just announced in August.
If you get this magazine, he has a big article in here. That's where I got it from.
And who do we have in 2 weeks?
All right. Cardiomyocytes, Aaron's
Yes.
humans, chemical and biological, and scaling up human pluripotent stem cells.
Yes.
All right. So those are the people to follow you.
Oh, okay. Gotcha.
All right. Rebecca is from the Great Lakes Bioenergy Center. She works with John Ralph, who's in both biochemistry and the Wisconsin Energy Institute. Rebecca, where were you born?
I was born in Winnipeg, Manitoba, Canada.
Manitoba. Way to go. And where'd you go to high school?
I went to high school at, in Winnipeg, in the south end of the city, Fort Richmond High School.
And where'd you go for undergrad and what did you study?
I went to the University of Manitoba for undergraduate studies, and I studied botany. I did my honors in botany. Main reason for going to that university is that my father was an English professor at the university, so it just made sense, and I could stay at home and literally walk to school. So it
That
was
is
great.
fabulous. What was his area of scholarship? Was it English?
He actually mainly studied theater and theology. It's an interesting
Two
combination.
THs.
Yeah.
Wow.
Yeah,
Excellent. And then where'd you go for your advanced degrees and what did you study?
I went to the University of British Columbia for graduate school. I just went directly into the PhD program. I studied with Dr. Lacey Samuels. And again, my degree was in botany. Specifically, I study plant cell biology and biochemistry, and now I do a lot of analytical chemistry as well.
And then did you go from UBC to here or did you have any other places?
I went directly from UBC to here to start my postdoc with John Ralph. And after 3 years of a postdoc, then I was promoted in a sense to being a staff scientist in this group. And that's the position I have now.
And how many years is that total here?
7 years I've been in the States.
Okay.
Yeah.
So how do you like our mild winters?
Oh, yes, very mild compared to.
I know. Usually we joke with people who come from San Diego. So how do you like that ice fishing? So today you're going to talk with us about cell walls and one of the more interesting things on the planet for people like me that study botany also.
Oh, good.
Thank you very much for coming to Plato.
Oh, well, thank you for having me and giving me the chance to present my research. I'm always happy to talk to people about what I do. So today I'm going to talk about the identification and characterization of plant enzymes to modify and add value to the plant cell wall, which is very descriptive, but also a bit of a mouthful. We can simplify it down to how to build a better optimal biofuel crop. That's really what we're trying to get at. So I'm going to start first by giving an overview of the Great Lakes Bioenergy Research Center and what we're hoping to do as a center and what our main goals are. And then I'm going to talk to you a little bit more about the science of creating bioenergy crops with lignin. If you have questions at any point during the talk, just let me know and stop me. It's easier to ask a question in the moment than having to wait till the end.
Thank you.
So Great Lakes Bioenergy Research Center is home to about 400, maybe more researchers at all different levels. We have faculty, academic staff scientists such as myself, postdocs, graduate students, undergraduate students, technicians, and we're all working towards the common goal of turning plants into fuel. The UW-Madison is not the only university that is involved in this endeavor. We also partner with Michigan State University, which is one of the other head universities. And we also have other universities across the country that are part of this: Texas A&M, Princeton, and also University of British Columbia in Canada. And we have partners all over the world as well as we continue our research. For those scientists that are part of GLBRC, at UW-Madison, we're located in the Wisconsin Energy Institute, which, if you're familiar with campus, is right here at the junction of Campus Drive and Old University. And relatively speaking, it is a newer building on campus. It's about 10 years old. And it was designed with energy efficiency and clean energy in mind. So it houses the researchers that are involved in GWRCs. We have plant biologists such as myself. We have organic chemists. We have ecologists, microbiologists, and bacteriologists. But we also have engineers that are interested in other types of renewable energy. So we have people who are working on solar power, wind power, nuclear power that are in the center and also affiliated with the center as well. So it's a very diverse group of scientists coming together, and really nice to hear about the different types of renewable energy research that are going on there. Our goal as a GLBRC is to work to develop economically viable, environmentally sustainable biofuels and bioproducts from dedicated energy crops grown on marginal lands. And I'll pick apart some of these phrases that are highlighted in green as I go through this introduction to explain to you a little bit more what we mean by that. GLBRC is one of 4 bioenergy research centers that is currently funded by the Department of Energy. The other ones are the Center for Advanced Bioenergy and Bioproducts Innovation, which is mainly centered out of Illinois State University, the Joint Bioenergy Institute, which is out in California, and the Center for Bioenergy Innovation, which is mainly centered out of Oak Ridge National Labs and also partners with NREL in Colorado. The push behind the Department of Energy into putting a lot of money towards biofuel research comes from this global climate crisis that we are experiencing that continues to evolve. And the fact that the amount of carbon dioxide that is being emitted into the atmosphere is causing global temperatures to increase and causing these major climate problems. And about half of the US energy-related carbon dioxide emissions from the last year were put into the atmosphere because of petroleum use. So if we were able to cut out our use of fossil fuels and switch to a more renewable source of energy, we could have the amount of carbon dioxide we're putting into the atmosphere, which would be ideal. The question we're asking, therefore, is can we make fuels and chemicals from plants, which is ultimately going to remove carbon dioxide from the atmosphere, because plants are incredible carbon sinks, instead of using fossil fuels, which is, of course, going to continue to add carbon dioxide to the atmosphere. And it's important to note that I'm mentioning not only fuels here, this is the fuels that are going to fuel our planes or our cars, but I'm also mentioning chemicals, because through the generation of fossil fuels and using fossil fuels, we get a lot of chemical byproducts that we use commonly in everything as a result of fossil fuels. We need to make alternate chemicals that don't come from the fossil fuel process, so that we create a sustainable, not just biofuels and bioproducts, but a whole bioeconomy that can be put into place instead. This is our ultimate goal, creating carbon neutral biofuels, where the amount of carbon dioxide we emit into the atmosphere is equal to the amount of carbon dioxide that we are pulling into the plants as the carbon sinks. Even better is if we can make this process carbon negative, so that the amount of carbon dioxide being pulled out of the atmosphere and being stored by plants is greater than the amount that we are emitting. Biofuels are not a new idea, as some of you might know. Back in the 1920s, in the New York Times, there was an article published that said that these types of fuels were the fuel of the future. It's going to come from fruit that is like sumac by the road, or apples, or weeds, or sawdust, or almost anything. So any vegetable matter, it can be turned into a fuel. You can just ferment it, stick it in your car. And in the 1930s, this is a photo showing that corn alcohol or ethanol was added to gasoline, something that's still done now. The problem with these types of biofuels, and this is the type of biofuel that typically pops into people's brain when they hear the word biofuel, is that it is using most often the fruit or the crop part of the plant in order to make the fuel. This is because that is where the starch is found in the plant, or the simple sugars that can easily be broken down to make a fuel. The problem with that is that you then have a competition between plants that are grown for agronomic purposes to make us food, and plants that we're growing for biofuels. These first generation biofuels are produced from biomass such as corn and sugarcane. There's a lot of corn ethanol that's being produced. This ethanol is primarily a gasoline additive. Biodiesel starch is primarily a biodiesel or a diesel additive. And as I said, it's typically coming from the grain of the corn and then being fermented to make ethanol. What we would like to do instead is use a different part of the plant, to use the lignocellulosic biomass to produce energy. So this is not the fruit part of the plant. Instead, it's all the other tissue, the stems, the leaves, everything else that is crop residue, or in the case of poplar, it's the stems and the leaves, all the biomass. Same for switchgrass, perennial grasses, and for sorghum. This material is highly abundant. The US produces over 1 billion tons of this material a year from agricultural residue or from forestry or from other acreage. And so if we can take that material, instead of just having it be waste, turn it into a biofuel that could push this our bioeconomy forward. Our bioenergy crops of interest include sorghum, perennial grasses, switchgrass, and poplar. You'll notice that corn is not on this list. That might be a little bit surprising, considering we're in the middle of the corn belt and there's a lot of corn being grown here. But the reason for that is that corn does not really hit our ideal characteristics of a bioenergy crop. We are looking for plants that are native to specific areas. If they're native to an area, we don't have to introduce an exotic plant into an environment. There's no chance that it will become invasive, which is important. We want as much as possible for the plants to be perennial. So the perennial grasses, switchgrass, and poplar all meet this characteristic of you can plant it, it will grow for a number of years before you harvest it. In those years, it's going to sequester carbon and remove carbon dioxide from the atmosphere and generate biomass that can be used. Sorghum is not a perennial crop. It is an annual crop, as some of you might know. But we are not interested, I should make this distinction, we're not interested in sweet sorghum or grain sorghum, which are used for food. We are interested in a type of sorghum called bioenergy sorghum, which instead of putting all of its energy towards making starch and simple sugars, is making a lot of lignocellulosic biomass instead.
Was it the same sorghum that farmers had used for silage?
That is a good question. No, I think that is forage sorghum. So I think it's a different type of sorghum.
So there's 3, forage?
There's 4 actually.
Oh, cool.
Yeah.
What are they?
Grain, sweet, forage, and bioenergy. Yeah. So this sorghum, you can see there's a person standing right here. This sorghum, it's not finished growing. It can be about 8 feet tall. So we produce a lot of biomass a year. The reason why sorghum remains on the list, even though it's an annual plant and not a perennial, is because it is so productive, and also because it requires very low input for it to grow. It's very drought tolerant. So as temperatures continue to increase and we experience more and more droughts across the country, this is a bioenergy crop that could be put into place and grow in a lot of those areas that are drought bitten. It also requires a lot less fertilizer input than corn. You don't have to be giving it as much nitrogen and phosphorus in order for it to grow. Therefore, it doesn't deplete the soil as much. You don't have to undergo the crop rotations that you do with corn to replenish the soil.
You do not have to rotate it at all. You can just grow it 10 years in a row.
As far as I know, but I am not a farmer, so
Okay.
I don't know as much about it. Yeah. We also are interested in growing plants that can grow in a polyculture. This is right here is a perfect example of a monoculture out at the Kellogg Biological Research Station at Michigan State University. And grown for research purposes, this makes sense. We're more interested in what you can see in these perennial grasses, for example, where you have a mixture of plants growing all together. That diversity of plants then extends to the diversity of the entire environment those plants grow in, into the biodiversity of the soil underneath, and the complexity of the microbial environment and the fungi that associate with the plants that helps ensure that the soil is healthy and that the environment remains healthy for these plants to continue growing. All of these ideal characteristics are leading to sustainable practices and being able to grow these plants for years and years and years, generations after generations, as bioenergy crops.
None of those plants are nitrogen fixing, however.
They are not, no.
Okay, that would be nice too.
That would be nice. We're working on it. We have some scientists that are, for example, trying to figure out the genetics behind the nitrogen fixation so that we can have sorghum that associates with bacteria and fixes nitrogen and puts nitrogen back into the soil. Yeah. So in the GLBRC, as I said, our mission is to generate knowledge needed to sustainably produce specialty biofuels and bioproducts from lignocellulosic bioenergy crops. And I fit in this first step in looking at these energy crops and determining what do these energy crops look like? What does their lignocellulosic biomass look like? Are there any changes that we want or need to make to it to improve these steps down the line where we're taking apart the lignocellulosic biomass and then making fuels from it? So let's take a closer look at the plant cell wall, which is where the lignocellulosic biomass exists. So if you picture your favorite bioenergy crop, I'm going to picture a poplar tree in my mind, and we take a cross section of it, and then we look at it under a microscope, we will see the individual plant cells. Every plant cell is surrounded by a cell wall that is found outside of the membrane of the cell. That cell wall is composed of three main components that are of interest to us. These are cellulose, hemicellulose, and a lignin. Cellulose and hemicellulose are both complex sugars. Lignin is in a different class of compounds called the phenolic. Cellulose makes up the backbone of the cell, or backbone of the cell wall, and it is made up of individual sugar molecules, primarily glucose, or actually only glucose, that are linked together through regular and specific linkages. It's a beta-1,4 linkage for those of you out there who really like chemistry. Those chains form and then they stack together to make cellulose microfibrils, as shown here, and then the microfibrils line up to form the backbone of the cell wall. The synthesis of glucose is really regular and it is regulated by enzymes that are involved in this process. So you end up with a very predictable structure, and you also end up with a complex sugar, but one that's very easy to break down. If you have one enzyme that can break that beta-1,4 linkage, then you can take apart cellulose. And breaking apart the cellulose leaves you with glucose molecules, which is a sugar, which can be used to make our food. The hemicellulose is another type of sugar, complex sugar, similar to the cellulose in that it often has a glucose backbone, but it has other sugar molecules that are branched off of it and integrated into the polymer. Again, its synthesis is regulated by enzymes, and so it has a very regular pattern, and we can predict what the hemicellulose will look like in specific plants. These hemicelluloses coat the cellulose and then bind the cellulose microfibrils together. So this just ensures that there's a little bit more structure to the cell wall. The last component of the cell wall is the one that I'm going to talk about most and focus on for the rest of the talk. This fascinatingly complex polymer, as I said, is a phenolic polymer, and its job is to basically find all of the spaces, all the gaps in the cell wall, and fill them and make the cell wall as rigid as possible. You can tell how many cells have a lignified cell wall in a plant based on the strength of the stem. So if you picture a tree, the majority of cells in a tree have lignin in their cell walls. That's why lignin is so strong. Lignin is what makes wood as strong as it does. If you compare that to a herbaceous plant like switchgrass, switchgrass has far fewer cells that have lignin in their cell walls. As a result, it's a more flexible stem. So the purpose of the lignin is to provide structural support to the cell wall, and that's also going to provide structural support to the stem. It allows stems to maintain their upright habit. Its presence in the cell wall also allows for water conduction over long periods, over long distances through the plant body against gravity. So for a plant, it's absolutely essential. If you pull all the lignin out of the cell wall of a plant, the plant will die. If you pull half the lignin out of the cell wall of the plant will be dwarfed and or it will fall over. It won't be able to maintain an upright growth habit. For these reasons, lignin is absolutely essential. But when it comes to processing the cell wall, And by this, I mean, we want to be able to take the cell wall, pre-treat it with enzymes or with chemicals and break it into little pieces. Lignin is recalcitrant to that. It doesn't like to break apart at all because that defeats the purpose of why it was put into the cell wall. What I aim to do and what the Ralph Lab aims to do is to look at this lignin and see how can we modify it to make that pre-treatment process easier so that it won't negatively impact plant growth or development. The plants will still grow normally. They will still have the lignin they need to grow. But when we get to that processing step, the enzymes will have greater accessibility to the sugars in the cell wall, and that lignin will just come apart a little bit easier. So that is what we are aiming to do.
Is this research new to the bioenergy efforts now over the last 15 years?
Yeah, I think the first publication where with the lignin modifications that I'll be talking about was published back in 2014. So it's definitely happened in the lifespan of the GLBRC. To understand how we can modify the lignin, have to get into a little bit of how it is made, and I'll try to make this as painless as possible. The lignin monomers, or they are called monolithic alcohols, as shown here, are synthesized in the cytosol of the cell. They are then exported from their site of synthesis in the cytosol into the cell wall. In the cell wall, they encounter enzymes, oxidizing enzymes like peroxidases and lacases, which turn these molecules into free radicals. That makes them really, really reactive. They basically have an electron that is bouncing around through these double bonds back and forth and back and forth. And so when it encounters another free radical, so another one of these monomers or a growing lignin chain, it binds to it. What this means, though, is that there isn't an enzyme that is dictating how the structure forms. The structure is forming solely based on chemistry, based on what monomers are here, based on what bonds they are most likely to form, what's most energetically favorable. As a result, you can get whatever monomers are present in whatever combinations they're capable of making. This just adds to the complexity of the molecule because you don't know what order these monomers are going to be in, what bonds will be there. And then added to that fact, these are not the only monomers that are part of lignin. Plants produce a lot of other compounds that are similar to that are toxic to the cell contents. And those ones which they cannot store in the plant vacuole are also going to end up in the cell wall. If they can also be acted on by these oxidizing enzymes, they could also end up in the lignin polymer. So as our methods for detection continue to improve, we're still finding more and more things that are ending up in the lignin polymer and adding to its complexity. As a result, the lignin that you see differs not only from one plant to the next, but can differ in different tissues in the same plant, and can even differ in cells that are sitting directly adjacent to each other. Because we don't have a good model of what the lignin looks like, it can become really difficult to try to modify it and find the most efficient way to modify it. We've come up with a few ideas though, and we call these designer lignins. The ones I'm going to focus on today are introducing novel chemically labile linkages into the lignin backbone. That is, bonds that are chemically weaker, it's not going to add weakness to the cell wall, but chemically they are weaker, and that's going to basically act as a zipper in the lignin, something that we can just open up and make the lignin fall apart a little bit easier. And I'll also refer a little bit to what are called value-added lignins. This is something else we're focusing on in the lab. This is the idea that because lignin becomes a bit of a dumping ground, if you can add compounds to the lignin that are valuable and are commodity chemicals and are compatible with the polymerization mechanism, when you are making your biofuels and you have your waste stream, because lignin tends to end up in a waste stream, if you're able to pull those chemicals out of the lignin waste stream, you're adding value back into the whole process. So instead of just burning the lignin as waste, instead you can get some money back out of it, and that can hopefully improve the economics of the entire process, because there's a lot more steps to the generation of biofuels than there are to making fossil fuels, unfortunately. So what is a zip lignin, or a lignin that has a zipper in it? This is a lignin, as I said, that has a weaker bond, a chemically weaker bond in the lignin. And the professor that I worked for, John Ralph, decided that the best bond we could introduce into the lignin backbone is an ester bond. This one right here, with the double bonded oxygen, this oxygen here. The reason why this bond is best is because it will break under mild base at room temperature. This is in contrast to the most typical bonds that are found in lignin. Most of those bonds require really strong acids or really strong bases and temperature in excess of 200 degrees Celsius in order to break. That's not ideal, nor is it safe. It's not energy efficient. It's very costly in terms of chemicals. And imagine having to work in a place where you have boiling sulfuric acid to break apart your cell wall material. So if we can use a mild base and room temperature instead, it makes the process more energy efficient and safer as well.
And when did we discover that break, that base?
That the mild base breaks.
That you could split it at that point. Was
Oh.
that known for a long time?
Yeah, it's been known for a long time that ester linkages are pretty chemically labile, that you just
Okay.
2 molar or 0.2 molar sodium hydroxide and they will fall apart. So the idea is that if we have these linkages and they are found throughout the lignin polymer, when you put it under these conditions, the polymer will unzip at those places. And what that's going to do is that it'll increase the accessibility to the underlying sugars. And as a result, you're able to get your enzymes or your other chemicals to the sugars and break them apart a little bit easier. The problem with this is that we have to figure out how we're going to put the zips into the lignin. We have to do it in a way that is compatible to plants. So we have to use known pathways or components that the plants can synthesize. And we have to use components that are actually compatible with lignification, because if they can't undergo that polymerization process, there's no way this is going to work. So thankfully, we found a group of enzymes that seem to be a good candidate, and these are the BAHD acyl transferase enzymes. This is a large family of enzymes in plants. The BAHD is an acronym that stands for the first 4 enzymes that were identified in plants that have this activity. And one of the activities of these enzymes is to take a CoA thioester, as shown here, and couple it to an alcohol acceptor and generate an ester-linked product. This is the product that we want, and we want that to be in the lignin. So we need to make sure that this would be compatible with lignification and that these two, this donor and this acceptor, are something that can be made by the plant. Luckily, we didn't have to look any further than the lignin biosynthesis pathway in order to find this donor and acceptor. I won't walk you through this pathway. A lot of it is on a need-to-know basis, let's just say. But these blue circles down at the bottom, this is one of the monolignal alcohols, and this is the other. These can act as our alcohol donors. And if we look a little bit further up in the pathway to intermediate, we have ferrulyl-CoA, which is a CoA thioester. So all we need to do is find an enzyme that can couple this ferrulyl-CoA to one of these monolignal alcohols and generate these products. And again, there's the ester linkage here that we'd be hoping to integrate into the lignin polymer. The reason why these zip conjugates, as we call them, would be ideal is that anywhere you see these arrows, these are places where these conjugates can be integrated into the lignin polymer. So the polymerization can happen at those points. And so both ends of the molecule can be integrated into the lignin backbone. So at this point, we needed to find an enzyme that would function as a ferrulyl-CoA monolignal transferase, an FMT enzyme. Luckily, we knew that all grasses couple p-fumarate the activity of an enzyme called a PMT. So we figured if there's a PMT enzyme, there must be an FMT enzyme out there for us to find. What we did is we started searching the literature, and this was done in collaboration with Curtis Wilkerson's group at Michigan State University. And we found that a plant called Angelica sinensis, or Chinese Angelica, has coniferyl ferrylate, one of our zip conjugates, in the roots at a level of about 2%. which per dry weight, it's a fair amount. This actually, they found it was not going into the cell wall. It was just floating around in the cytoplasm of the cells. But nonetheless, if it's producing this conjugate, it has to have the enzyme. So we were able to, Kurt's group actually was able to find the gene encoding the enzyme and then test the activity of the enzyme. And then we also put the enzyme, or the gene encoding the enzyme, sorry, into poplar because that's one of our bioenergy crops. So here are some of the major players involved. There is John Ralph standing in the greenhouse, showing you how tall the poplar trees were growing once we got the zip gene into them. Sean Mansfield and his group at the University of British Columbia were the group that put the FMT gene encoding the enzyme into poplar. And Greg Sanford was growing these plants for us in the greenhouse and helped us maintain and establish these plants.
How old is that poplar plant?
This poplar plant, I believe at this point it was maybe a year old.
It was what?
Maybe a year old.
A year or so?
Yeah.
Okay.
Yeah. And we also moved from at some point after this, moved from the greenhouse to field trials as well with these plants, which we were allowed to do for 5 years, I believe. Okay, so we know that the gene was in the plant. We know that the enzyme was active. What we needed to test next was, did we actually make a modification to the lignin? Did we put those zip conjugates into the lignin? And the way that we tested this was using a lignin chemical analysis method called derivatization followed by reductive cleavage, or DFRC for short. It is no coincidence that John happened to be working at the Dairy Forage Research Center when he and one of his graduate students developed this method, hence why he wanted to call it DFRC. The nice thing about this method is that it cleaves the most common bond, the beta ether bond in the lignin backbone, but it leaves these ester bonds intact. This means you get a distinct chemical fingerprint for any compound that has that ester linkage. And we can separate out these compounds from any other monomers that are released from the lignin polymer. We can use gas chromatography, mass spectroscopy to identify the mass of this total molecule, and then also find its specific fragments. Long story short, what we saw in the end was this green peak here indicates, is showing the presence of these zip conjugates in the lignin polymer. And because we're able to release them through this DFRC method, that means they were integrated into the polymer, not necessarily just pendant off the chain. So this was really exciting. It means that our idea was working. However, it doesn't mean much if it doesn't make any changes to the digestibility of the cell wall. So that was the next thing to test. What we did is we ground the poplar material and then we pre-treated it with the vial base at room temperature. And then we exposed it to enzymes that would break apart the sugars in the cell wall and release glucose. And with the improved glucose yield, that means that there was just more accessibility to glucose and you can pull more glucose out of the cell wall. So we had a number of lines where we had significant improvements on glucose release compared to the control poplar tree. This indicated to us, well, this method seems to be working. If we can increase the amount of zip conjugates in the cell wall at least a little bit, well, then we can increase the digestibility. So this seems to, this showed a lot of promise. One of the things that we noticed through this study, though, was this small little peak in black right here in the control poplar line. This is found at the exact same place and has the same chemical signature as this zip conjugate peak. This made us a little bit concerned. And we thought at first, surely it's just contamination. So we cleaned all our milling jars, we changed out all of our solvents, we changed the column on the GCMS. We tested a number of different poplar samples and we ended up with the same result every single time. There's a little peak right there. And we could only come to the conclusion, oh, poplar trees naturally have some zip conjugates. Mother Nature got there before us and went, this will be a good idea. So this led to a study that was headed by my co-worker, Dr. Stephen Carlin, in which he decided to look at the entire plant kingdom or representatives throughout the plant kingdom to determine how prominent is these native zip conjugates throughout lignin. We looked at gymnosperm species and angiosperm species Gymnosperms, in all the examples we looked at, we never saw any zip conjugates. But we saw a number of angiosperm species, including camelonids or grasses, and species in the rosids, such as poplar, knaf, and balsa, which did produce zip conjugates. This indicated to us sometime between the evolution of gymnosperms and angiosperms, you end up with these zip conjugates in the lignin and the cell wall. But it's not necessarily consistent. You have a lot of species that have them and then species that are closely related that don't. What was particularly interesting to us is that some of our bioenergy crop species of interest, switchgrass, sorghum, even maize, have these zip conjugates. In fact, every single grass that we tested have zip conjugates in the lignin. So somehow that is evolutionary beneficial for these plants. This opened up possibilities for us because we recognize that these natural zip levels are quite low, even in sorghum, because when you compare it to the other things that are present in the lignin, the total composition of the lignin, the zip conjugates make up a very small percent. But if we could increase them further, we would increase the digestibility of the cell wall further, based on what we saw in poplar. But it also introduced the possibility of there are a lot more FMT enzymes out there that we haven't identified. And if any of these FMT enzymes are more active than one we already have, we could be producing more conjugate into the cell wall. That could be great. So what I did is I looked at the one FMT from a grass that we knew, which is an FMT from rice. And then I looked at the amino acid sequence, and from that, identified the most likely candidates for FMT enzymes from sorghum, switchgrass, and maize, which are located, it's these three right here that I was studying, and tested their activity. For this, we collaborated with Dr. Brian Fox's group in the Department of Biochemistry at UW-Madison. And the reason that we worked with him is that his group does a lot in a system called a wheat germ cell-free translation system. Typically, when you want to test the activity of a protein, you put your gene into a bacteria, you get that bacteria, like E. coli, to make a lot of protein, you purify the protein, then you test its activity. Acyl transferases don't really like that. Once you get to the purification step, they tend to pellet out and you can't get them soluble, and so you can't test their activity. But in the wheat germ cell-free translation system, what you're doing is you're taking all the protein biosynthesis machinery from wheat germ, in short, simple terms, basically putting it into a test tube. You put your gene of interest in there, the protein biosynthesis machinery turns it into your gene into the enzyme, and then you can add your substrates right into that test tube and you can test its activity. So this is what we did. I fed it the monolignal alcohols, and I fed it frulyl-CoA as the CoA thioester. And the pink and purple peaks that you see here are showing that we were able to make zip conjugates in these enzymes. This one is that first FMT that we identified from Angelica sinensis. This is the rice FMT, maize, switchgrass, and sorghum. So you can see here, at least based on activity, this rice FMT is really good. It's very, very active, produces a lot of product. The sorghum FMT kind of falls second. Then the switchgrass FMT, and then maize FMT is not terribly active. These test tube enzyme tests then led us to decide we need to test these enzymes also in plants. And my go-to plant for testing enzymes, testing genes of interest to begin with, is Arabidopsis Thaliana. This is not one of our bioenergy crops, but it is a good model plant. Grows very quickly, produces a large seed set, and genetically we can do just about anything to it. It's very easy to put genes into this plant. So when I put these FMT genes into the plant, this is a wild type plant or a control Arabidopsis plant, and this is a plant that has the FMT gene. As you can see, they look the same. So we have not changed the morphology or growth or development of the plant by adding the gene. I also looked at where the enzyme is active, and this green fluorescence here is showing where the enzyme is active. The red dots are chloroplasts in the stem. So these are stem longitudinal sections. Picture a stem, longitudinal section, and I'm showing the cells. The green fluorescence shows the cells where this enzyme is active. This is where we want it to be active. A lot of these cells are lignifying cells. And most importantly, this enzyme is active in the cytoplasm of the cell. It's not in the cell wall. It's not in any of the organelles inside the cell. This is very important because that's going to be active at the same place in the same time as the lignin biosynthesis, which means that the substrates we need for this enzyme are going to be right there. Next, I use the DFRC method to determine if we actually were able to get those zip conjugates into the cell wall. And we have these ester bonds in the cell wall. As you can see here, this pink peak is just showing the amount of zip conjugate that we were able to get into the cell wall, indicating all these enzymes are working not only in a test tube, but they're also working efficient in the plant. This has led to where I am now, which is putting these FMT genes into sorghum and trying to increase the FMT activity and the amount of zip conjugates in the lignin. And this is not only to potentially further increase the digestibility of the cell wall, but also those zip conjugates, when they are in the lignin waste stream, you can release ferulic acid from it. And ferulic acid is one of the commodity chemicals we're potentially interested in. So we have our energy crops and with the poplar or with the sorghum. And then the next step is I hand this material over to researchers that are then going to take these, take the plants that I generated, and they're going to break them down into the component parts. Hopefully a little bit easier and using less energy because I've modified the lignin. They will do this biomass breakdown using heat or acids, pressure, ammonia gas, salts. There's a bunch of different chemical methods you can use for it. And they'll also do it using enzymes that will break down the cellulose into the glucose molecules. Once we've broken down the material, both the lignin and the sugars, then is the fermentation step. And this is where we hand it off to yet another group who have engineered microbes, bacteria, and yeast that can eat the glucose and turn it into fuels such as butanol, and that can also digest some of these lignin compounds a little bit further and turn it into, for example, alternative plastics. And that leads us to the production of biofuels and bioproducts and completes our pipeline. Some of the challenges we face is, of course, make sure energy crops that we're using. Energy, are we chemicals we use? Also, economics is a huge challenge. There's so much that is cost system to compete. bioenergy, a way that we didn't affect any of the agronomic traits, but we also improved the processing. But then in the last 5 years, Department of Energy decided to deal with their energy crisis.
Seems to me that nuclear is such a much easier answer.
Yeah, I think it's the safety issues behind it, you know, the fear that people have over it.
The fear that people
Yes.
have, but it's not real,
Yeah.
I don't.
And what to do with the waste. So I was there when the moratorium was lifted on, the announcement was made that the moratorium was going to be lifted on nuclear power research in Wisconsin. And the question was asked by somebody, what are you going to do with the waste? And the answer, unfortunately, was, well, other states have dealt with it, so I'm sure we'll figure out a solution, which is kind of unsatisfactory. So I think it's the waste and it's the potential environment repercussions of nuclear power.
Well, I understand that the new smaller nuclear fission processes use up most of the energy waste. Like now we're accumulating all this waste, but the new nuclear procedures, processes actually use the most of the nuclear waste. You don't know.
I don't know, I'm sorry. Yes.
They are harvesting popular, like quaking aspen,
Mhm.
differently now. The growth cycle is shorter,
Mhm.
and they're harvesting this particular crop more often. And they're clear cutting
Mhm.
as much as, well, I've seen 20-acre plots,
Mhm.
and they rotate this. And they do all of the chipping on site because of the way they are harvesting it.
Mhm.
It's almost like harvesting corn almost,
Mhm.
but they have all of this material for producing.
Yeah. We can use basically anything that's left over. If there's chipped material that's just kind of sitting around, that's still lignocellulosic biomass. We could still work with it and put it through our fermenters.
Because they're just harvesting these trees for long fiber,
Yeah,
for paper.
yeah, you know, things that are left over, we could definitely use that. Yes.
Yeah, now, when you got the lignin growing with these little free radicals, that
Mhm.
you had, so and then you want to increase the number of these little,
Mhm.
so they, the radicals that grab onto those periodically, is it just kind of like random, like the concentration of it there that determines how that'll happen, or is there a way that maybe it could be encouraged to be in the neighborhood and
Yeah,
get grabbed?
I wish there was some encouraging factor. We can sort of dictate the timing for when the enzyme is active. That's about as close as we can get. Other than that, it's trying to overload the system with making these zip conjugates, just so as many of them as possible will be in the cell wall. And because the zip conjugates use the monolignals as well, that will also try to push the system a little bit away from making just monolignals to making more of these conjugates, and the more the conjugates will be in the cell wall. And then in theory, they have to become part of the conjugate. The tricky thing that we run into is that sometimes if you have too many conjugates in the plant doesn't really like it. So there's a maximum threshold that we're still trying to determine where exactly that is. And if the plant doesn't like it, it has mechanisms to just shut things down and say, I'm going back to business as usual. That enzyme's not going to work in here anymore. So one of the complications of working with the biological systems.
And are you working with switchgrass as well as popular?
I don't do as much work with switchgrass. There are other groups that do. We had, for the first 10 years of GLBRC, really well-established breeding programs for switchgrass rather than generating genetically modified plants. And I think that's been switched over, and now we have groups that are working on changing the flowering time of switchgrass, because that can impact how much biomass is being produced by the plant. But personally, I haven't done any switchgrass research.
Anybody else?